the PowerPoint.
Say it again. I didn't quite hear what you said.
Yes, for this chapter, chapter 17, I couldn't get into the PowerPoint slide that you have.
Oh,
It's
that's
like require access, so I don't know where can I find it.
like you want to be in it right now while we're talking?
Yes, I have a couples like other like devices, so I can like, look at it at the same time that you're sharing your
Hold on.
screen. Yeah, but as the link that you posted on, we couldn't like I couldn't get in.
Can you open it now?
Yes, like
Try it now.
yeah. Thank you.
It
Okay.
did that to me too, but the only reason why it was doing that is because I wasn't logged into my school Gmail. You have
Yeah,
to make
that's
sure
what
you're logged out of everything and only logged into TCC.
Yeah, that's exactly right. That's what happens. I get those emails, and that's why I don't respond to a lot of those because I get them so much. But Marissa, just to answer your question, we're doing the DNA stuff today too. So we're doing both. So you are prepared. Okay. So like I said before, we can't use antibiotics to treat viruses. Typically, our typical antibiotics attack bacteria in two ways. Well, there's more than two ways, but two of the main ways that you are familiar with that we've talked about is they attack the bacteria's cell wall synthesis. So remember, what do bacteria cell walls, what are they made of?
Peptoglycan.
Peptidoglycan. So if we block that, remember, if you take away a cell wall from a bacteria, it typically doesn't make it. Or the antibiotics target the ribosomes of bacteria and block the central dogma. Well, viruses don't have peptidoglycan and they don't have ribosomes, so that's not a good method to treat viruses. That's why antibiotics don't work when you have a virus. So instead, we can use antivirals, and we'll talk a little bit about those later, or vaccinations, right? Those vaccinations are basically preparing your immune system ahead of time to be prepared for an infection from the virus so that you are capable of fighting it quickly once you are exposed to it. So when we make vaccinations, we use parts of the virus so that our immune system can recognize it and develop a primary immune response. So this question says, what part of the virus do you think it would be important to use to make a vaccine so that our body can recognize it and destroy it? Do you think it's the protein part of the virus or the DNA part of the virus?
Protein.
That's right. We're going to make it
so it never connects.
Right, exactly. So that it never connects. That's exactly right. And so that when we recognize it and it's on the surface, we can recognize it very quickly and get rid of it. Okay, when we look at viruses, we classify them in a variety of ways. You've already seen that they come in all of these different shapes and sizes, and so that's one way they can be classified. A more common way is whether they have DNA or RNA, and then whether that DNA or RNA is single-stranded or double-stranded, and then whether it's linear or circular. And viruses don't have the same rules that cells do, so they can kind of come in all of these different shapes and sizes, and it's okay. Like you can have a double-stranded RNA virus, for example. So just kind of showing you some of the different ones again. I think they're kind of neat looking. They're kind of pretty in a sense. I know that sounds weird. All right. So viruses aren't living. Instead, they live kind of what's called a borrowed life. They have to use the machinery from their host cell to do the work that they need to do. So the host cell will provide ATP. The host cell will provide the ribosomes. The host cell will often provide the enzymes necessary to make new copies of the DNA or to transcribe the RNA, to translate the protein. And then the proteins that are made from the genes of the virus are typically the proteins that the virus needs to reassemble itself within a cell. All right. And viruses are very, very specific. They don't just, willy-nilly attack any cell. They very specifically recognize receptor proteins on the cell surface. And so that's why we have respiratory viruses that are a little bit different than, like a sexually transmitted virus kind of thing, right? So those are different kinds of cell types, and so they infect different things. Now, one of the things about the coronavirus, it infects cells that have that ACE2 receptor. Well, that happens to be on more than just the cells of your respiratory system. So you may have been hearing that one of the symptoms is gastrointestinal. So people are getting more gastrointestinal issues from this as well, because that ACE2 receptor is a little bit more broad ranged and not just on that kind of cell type. Okay, so let's just ask this concept check question. Which feature is not found in all viruses?
C.
Good, the envelope. So some do, some don't, but they all have the genetic material within a protein capsid. That part is true on all viruses. All right, so before we talk a little bit more about coronavirus, I want to go through how viruses basically infect a cell and make more of their own virus. All right, there's 5 steps. First, the virus has to recognize the cell and attach to it in some way. So it's those surface proteins that are attaching to the receptor proteins on the cell. Then the virus has to somehow penetrate into the cell, and that happens in different ways, and we'll talk about different ways that this can happen. The way the coronavirus works, and this example on this slide works, is that it happens through receptor-mediated endocytosis. So we talked about that when we talked about active transport. All right, so it has to bind that receptor for that endocytosis event to happen. Then once it gets inside the cell, it releases its DNA or RNA out, and then uses that DNA or RNA to kind of commandeers the cell and says, hey, we need to make RNAs from the DNA. If there's an RNA from the DNA, I don't know what just happened, sorry. We need to make proteins from these RNAs. We need to copy these RNAs. We might need to make DNA. So it goes through a process of transcription and replication and translation. It starts to build the proteins that are necessary, and then the viruses basically reassemble inside the cell, and then they basically cause the cell to burst, and they release the viruses out. And you can make a lot of viruses from one infection. So it depends on what kind of virus you are as to the steps that really have to take place. So, and this is kind of an easy example for us to envision, because the virus in this case has DNA inside of it. So the virus gets brought in via endocytosis. There's a moment where the virus envelope and proteins then kind of disassemble, releasing the DNA into the cell. Now that DNA is used for 2 purposes. One, we're going to go through replication, which is something we're going to talk about today, to make more copies of it to package up in the new virus. That DNA is also going to be used to do transcription and then translation to build the proteins that make this new virus, and then the virus will be released. Any questions on that general gist of how that works? Now, RNA viruses work a little bit differently. Obviously, if it's RNA, then they have to go through translation to make proteins, but they also need to make copies of themselves. So many times the way they do that is they actually go through a process called reverse transcription. So they actually take the RNA and they go back and make DNA from it, and then they make lots of copies of the RNA to package back into the new virus. And that enzyme that controls that reverse transcription is unique to viruses. We don't really have it in normal cells. And it's provided by the RNA has the gene for it, so that it makes that protein via the cell's ribosome to go back and then do that step.
You said that some have a double helix in RNA, so do the RNA split completely and make 2 new DNAs and RNAs?
I don't know all of the details of that. I don't know. I would think you would have to copy each strand, yes, but I don't really know for sure.
Okay,
All
sorry,
right, now,
just curious.
no, that's okay. And I'm not a virologist by any means, so you're going to ask me questions that are fabulous questions that I don't know the answer to, but that's great. They're great questions. All right, so what about the ones that don't have a membrane to how do they do it? And we're going to talk a little bit more about this one. So this is the, if you recognize it, the one that infects bacteria. So it kind of lands on the cell, recognizes some receptors, and then basically what it does is it literally bores a hole through the cell membrane, and then instead of having the entire protein capsid enter the cell, it only squirts its genetic material in. So it's just squirting the genetic material in, and the actual capsid protein part of the virus stay outside of the cell. And that's going to be important to us later. So you can kind of see the different routes that they can take. They can either get endocytosed like this, and that releases it, or they can just fuse with it and make a hole that then spews it in. So this is more like kind of exocytosis in a sense that it's fusing and then spewing stuff in, but this time it's going into the cell. Okay, let's skip that question. So the viruses, when they're infecting the cell, they can either have immediate effects on the cell or they can have it be delayed. If it's immediate, it falls into the category of being a lytic infection. So remember, we've learned the word lysis. That means to break. We talked about hydrolytic enzymes that break up molecules. So the lytic infection basically is what lyses the host cell, breaks it up, releasing the new viruses that are being made. All right, I'm going to come back to phage therapy in just 1 second. So here's an example. Here's the attachment of the virus to the cell. And this is the phage one, so you can see that it's squirting the DNA in. It's then assembling all of its new parts, making new viruses, and then causing the cell to actually burst and get rid of it. So you can imagine if this particular virus infects specifically bacteria or specifically E. coli, that this could be a possible mode of being an antibacterial for an infection, right? And so there is a lot of research on what's called phage therapy for bacterial infections, because the phage would attack just the bacterial cells and kill them. So you could have it target specific bacteria from the infection that you have, and only kill those cells, not harm any of the other cells around you. And it's still not something that's fully figured out and worked out. I think it has been used in a couple of cases, but I think the volume of cell death kind of lags behind how useful it is. So, but it's something people are studying. All right, so just another example. Here's, this is herpes. And, okay, so Juan just asked me a question. Sorry, if you say they can mutate, then they mutate to attack different cells. That is possible. So you've heard of how, you know, like the swine flu or the avian bird flu, right? Or even this coronavirus, they thought maybe originated in bats, right? So those obviously even went from not just one cell type to another cell type, but from one organism to another organism. So yes, it's totally possible that they could mutate and attack different cells. It's probably a little less likely to mutate to attack an E. coli and then all of a sudden be able to attack a human cell, because the cell surface proteins between an E. coli and a human cell are extremely different, whereas between a pig and a human, they're pretty similar. So you don't have to do a lot of mutation. But that doesn't mean that over a long period of time, which is how natural selection normally works, that couldn't happen. It's totally possible. So I like this picture here just because I want you to remember your cell biology, right? So it shows you how, you know, once it's made, it's kind of treated like a vesicle. It travels along a cytoskeleton, goes into the nucleus. It's going to use enzymes like RNA polymerase. It's going to use the cell's ribosomes. It's going to go basically kind of through the secretory pathway to get to the cell membrane to be sent out of the cell, and you make lots of new virus for just one infection from one cell. So that's how viral infections kind of grow so exponentially so fast. And you may have been hearing with the coronavirus that people seem to have, those that aren't making it, they have kind of minimal symptoms, and then very quickly it goes from they're having trouble breathing, and then they die like the next day. Like once they start having really serious symptoms, it's pretty quick. Now, part of that is because it's in your lungs and you can't breathe, right? So that's part of it. But it's also probably we're getting to a threshold level of viruses that no cell is making it, and they're all being attacked. So this is the COVID-19 or the coronavirus pathway. So it uses receptor-mediated endocytosis to get inside. It's an RNA virus, so it's going to insert its RNA. That RNA is going to go through translation immediately to be made into that replication transcription complex. So now it's going to make this protein that can do both replication of the RNA, so it doesn't have to go back to that DNA state, and transcription of this translation of this RNA, and it makes all of these different, and we're going to talk about this in the next chapters, not today, but next class. It's going to make all these different RNA versions that can be made into various different proteins that are part of the virus. So you can see they go to the rough ER, those proteins to be folded, and then they go to the Golgi, and then within the Golgi they're basically being made into those full virus particles that then are packaged up into vesicles and taken out to lyse the cell. So any question on the life cycle? So
When they
you might
enter the cell, are they are they going straight to the nucleus?
They do, yes. Typically the RNA, well, if it's a DNA virus, it definitely goes straight to the nucleus. If it's an RNA virus, it doesn't necessarily have to. It has to find the ribosome first, and then it can make, if it needs to go back into the nucleus, it will. But it kind of commandeers the cell and just makes those first couple proteins, then they just take over the cell altogether. So you might be wondering, like, what do I expect you to know? In general, what the big things I want you to know is the steps of infection. So those 5 steps that I showed you about attachment, penetration, synthesis, assembly, release, right? And then I also want you to know how we can classify them into different categories, meaning they can be RNA or DNA, and that really tells us a lot. And then I also think it's very important to know that they have very specific host cells. So we're going to talk about that in just a minute. So Marissa asked, do the cells die? Absolutely. The cells are dying at this point. All right. So when they typically, like that last slide, when they're releasing a whole bunch, and all of those are coming out of the cell all at once, and the cell is basically breaking up, exploding in a sense. Okay, now that's called the lytic infection. The other way this can happen is through another pathway called the lysogenic pathway. So if you kind of break that word, break it down, it's still going to lyse the cells, but not right away. And the genic part, maybe you can think about is it stays hidden in the genome for a little bit. So in a lysogenic infection, what happens is the virus squirts its DNA into the cell, and instead of immediately being replicated and transcribed and translated, it just cuts the host DNA and squirts itself in and just becomes part of the host DNA. So it's like Red Rover, it just joins up with it, right? And then because it's part of the host DNA, when the host cell replicates its DNA, it's going to replicate the viral DNA with it. So all the next generation cells are going to have the virus in them. All right. So this is kind of what happens with the wart virus that Ivan was talking about. And then when your body or your cell undergoes stress, so for example, probably right now is a stressful situation. So you might have a cold sore, or maybe you have a cold and you get a cold sore, or sometimes things flare up at odd times. Typically, that's some sort of indication that there's stress in the body. And when that happens, that virus DNA excises from the host DNA, kind of cuts up the host DNA up into pieces, and then commandeers the cell again, utilizes all the cell parts to make more virus particles, and then starts killing all of those cells all at once. Okay, so that's the lysogenic part. And it's lysogenic because it can go many generations without killing the cells, but in the lysogenic, you always have a little bit of the lytic at the end. So the lysogenic one still ends up with lysis, but only in those stress events. All right, so it's probably a good idea.
I'm sorry, how long
No.
can that cycle last then?
How long can it stay hidden? For years. It can be hidden for a very long time. So, for example, herpes, the one that causes cold sores, pretty much once you get it, you're kind of stuck with it for the rest of your life. There's not a lot you can do, but you only really have it flare up when you're in a stress situation. And so, therefore, you don't always see the results of it. All right, so here it says cold sores that form on your lips. The stress cells then start to release the viruses. That infects the neighboring cells. So the cold sore is actually the whole bunch of those dead cells kind of collecting. And when the cold sore is active and hurting, that means those cells that have the virus are still kind of sending virus out and killing cells. HIV is another one that kind of can sit and be dormant for a while. So you can be exposed to HIV and not necessarily have AIDS yet, because remember, HIV, we talked about this one earlier, it infects your immune cells, and AIDS is the result of having so little immune cells left. We talked about those helper T cells, I think. Did we talk about that in both my sections?
Yes.
Okay, so it infects those helper T cells, and so it's only when there's enough virus kind of starts to accumulate in the system, and your body kind of has a little bit of a stress response, that you get an active viral infection that's just kill those helper T cells, and you get full-blown AIDS. So people who have HIV can live for a very long time without any effects from it. I mean, nowadays you can live because there's lots of medicines for it to kind of help. It actually kind of suppresses your immune system a little bit so that your T cells remain capable of doing what they need to do. Human papillomavirus. So you all had to get an HPV vaccine to get into TCC, didn't you?
Yes.
This is the one. It's very, very prominent. Pretty much, if you're sexually active, you have it. I mean, it's very, very, very prominent. And so one of the reasons why they want you to get the vaccination now is because we know that most sexually active people are being exposed to this. You can at least help your body protect against it, because it does cause cervical cancer in women pretty easily. So having the vaccination against it, even if guys don't get cervical cancer, you're helping not spread it. You're not having a live infection that can be spread to a female. Okay, so how do we treat these things? So there are antiviral drugs that will basically target enzymes and other proteins that are pretty virus specific. You have to know a little bit about the virus. We do know a lot about viruses now. We've studied them quite considerably. A lot of times they also kill the host cell, so you have to take that into consideration. So for example, when you're treating a wart, you are not just killing the virus, you're killing your own cells as well. And then it gets complicated because not every virus is the same, so you can't just use the same antiviral for every viral infection. So, so for example, Tamiflu, it's really only effective if you take it when you have very little viral particles in your system. Once you're past kind of the 24-hour, 48-hour mark with the flu, Tamiflu is not very effective because it's just too much virus in the system for it to be an effective drug. The other thing we can do to protect us against viruses is vaccinations. I am totally pro-vaccination, and I'm not afraid to stand on my soapbox and say it. First and foremost, vaccinations can't give you generally don't give you the disease, and we'll talk about what that means in just a minute. So I want to just do a little minute on how our immune system works. So typically what happens is when we get infected with something, our immune system has a pretty drastic response to that infection. And then it has a memory capacity. So once it sees that infection, it remembers that we saw it. So then the second time our bodies are exposed to it, the immune response is quick and huge to fight against the infection. Let me see if I can find, I was going to pull this up and I forgot. Immune response. I like this. I'm on my my desk computer, my office computer in my house, and it's so much nicer because I can just change screens. Oops. Okay, so this will kind of show us it. So what happens is this. When we're first exposed to something, our body has this immune response. And then our body has memory. So during this kind of downfall from this immune response, our body is creating a memory of what we just saw. And so when we're exposed to it a second time, that memory kicks in and our immune system very quickly goes into high gear. Look how much higher the immune response is. And it's so much immune response and so fast that we don't even necessarily know that we've been exposed upon this second exposure because our bodies are fighting it so quickly. All right. So that's why we say you can't really get infected with something twice, because once you've been infected with it, once you've had a certain virus, then chances of you getting it again, that same exact virus, now it has to be the same exact virus, is low because your immune system's going to have a response to it. So that's like the idea behind vaccinations, is to promote that primary immune response with something that's not harmful to us, so that when we are exposed to the real virus or bacteria, we have that secondary immune response that happens really fast, and our bodies fight it before we even know we've been exposed. All right. So I just put some graphs on here to just kind of show you. So this was the number of cases of measles. These were the number of cases of polio. Measles, for example, is extremely contagious. If one person gets it, I think it's more contagious than the coronavirus. People around you are going to get it. And you can see that, lots of people had measles over these decades right here. And then they created the vaccine and started giving it, and look at the numbers of measles cases after that. So this is how effective, literally can wipe out that type of infection in a whole population of people. Same thing here. Here's polio, people with polio. So just kind of an aside, my grandpa, who was orphaned by his mom that had the Spanish flu, he ended up getting polio before there was a vaccine. So he had he had it in his right side. He had a limp right arm from polio. So they introduced the vaccine here, and you can see that polio is just eradicated. It's gone. We don't have it anymore. So this is why vaccinations are extremely critical, because they help protect everybody. So getting vaccinated, kind of like how we're talking about with social distancing, is a selfless act, because you're not just helping yourself, which you are helping yourself, but you're helping others around you as well. Because there are people who are immunocompromised, that maybe they don't have that secondary immune response as well as everybody else, Or there are people who maybe a lot of vaccinations are made in eggs, and so if you have a severe egg allergy, you cannot get vaccinated. And so those people kind of live every day at risk and have to depend on the people around them to be fully vaccinated. The other thing that's important to remember is our memory in our cells is not a forever memory. So it's oftentimes helpful for especially like older people to get re-immunized for things maybe that they were immunized when they were younger to stimulate that immune response again, because sometimes the memory, and people are living longer and longer, and we're seeing that immune memory isn't as long as our lifespan is becoming. Okay, so this is just a good comparison of flu shot versus no flu shot. So what this is the probability of getting the flu. These are both statistically significant in terms of their p-values. And then down here we have number of times getting the flu over 20 years. So people who get no flu shot have a much higher percentage of getting the flu once, twice, 3 times, 4 times, maybe even 5 times over a 20-year period. If you get an annual flu shot, most of the time your probability is to get no flu. Now some people do get the flu, and I can tell you that I've gotten the flu even though I've gotten the flu shot. But chances of you getting it twice with the flu shot is very low, right? If you get it every single year, right? And look, there's like no at 3, and there's no 4, and there's no 5 at all. Now let's talk about how these these vaccinations are made. So there's different kinds of vaccinations, and we're going to talk about these first 3 first. Live vaccines are situations where they actually give you live virus in really low amounts. So you can't do this with a virus that replicates really quickly or is super, super, super contagious, because that's dangerous. If it's a slower kind of developing virus, you can do this at really low amounts, create that primary immune response so that if you're exposed to it later, you don't get it. But it has to be extremely low amounts. And look, there's not a lot in that category that you've heard of, right? Some are what are called attenuated vaccines, and I feel like maybe we talked about this in lecture, but I'm just going to kind of tell you how this works again. So they'll take a human virus, for example, and they will infect, typically they use chickens. They'll infect chickens with the virus. Then they will extract the virus from the chickens, and they'll reinfect another chicken. And then they'll extract the virus from the chicken, and they'll reinfect another chicken. Basically what they're doing is they're kind of trying to speed up natural selection. They're trying to cause the virus to mutate so much that it would be able to infect the chicken now and not humans anymore. So that's the whole idea behind it. Hopefully it's changed enough after many rounds of infecting the chickens that it's changed so much that now it's not going to be able to infect humans. All right, and in some cases that has been extremely usable. So even though you're giving a live virus to humans, because it's now changed, you're only giving it during the vaccination moment, and it's changed enough at that moment that your body's response to it is building the immune response, but you're not getting sick from it. So even if it were to mutate later, it doesn't matter. All right, so you can see there's a lot in here. These are oftentimes the ones that are given up your nose or orally in some way, okay? Probably the best category are these killed inactivated viruses and cellular fraction vaccines, because basically what these are doing is just taking little teeny pieces or the toxoids too, pieces of the virus, either surface proteins on the virus. Some viruses make toxins, so you can take those toxin proteins. And basically what you're doing is you're only putting in a little piece of the virus to stimulate that primary immune response. so that when you're exposed to the big virus, the whole thing, your body still recognizes that little piece that was put in. So maybe you only put in, for example, if you're thinking of the coronavirus, maybe only put in the spike protein. The rest of the virus isn't there. The spike protein by itself can't make your cells sick. But if your immune system recognizes that spike protein, then you're going to, anytime it comes into contact with your immune system, you're going to go into that secondary immune response. All right. And one of the things they're talking about now, which isn't on this, are basically taking antibodies from someone who's already been infected. So taking their immune system and putting into people that are currently sick and hoping that that's enough to fight the virus. So that's likely what's going to happen as we move forward with coronavirus. Is my guess is once the numbers start to really fall and they're thinking about whether people should go back out and risk this, you will probably be required, or I don't know, or you can opt in or opt out to get, first of all, tested to see whether you've been exposed. And so the way they do that is they just look for those antibodies in your system. So they look for your immune cells to make the antibodies to fight the virus. And then if you've been exposed, then chances are you can go back out and go to the store and you're fine, right? Because even if someone coughs on you, you've already been exposed. So you would probably have to be test negative for live virus in your system and show that you have the antibodies in your system. If those two things happen, then you're safe to go back out in the public because you've already had it, but you're not actively transferring it at the time. So my guess is that's where we're going to go until we have the vaccination out there. Well, they call that antibody therapy. They don't really call it a vaccine. It's called antibody. It's called passive immunity. So you're just basically, instead of stimulating my own immune system, I'm just passing on my immune system to somebody else and hoping it works. It's somewhat how it works between mother and child. That's a passive immune spreading. So the mom gives some of her immune system recognition to the baby as the baby's developing. Okay, so that's it on viruses. Do you guys have any questions on viruses before we move on to DNA? I don't have a good video on viruses. I was looking for one that was short and sweet and to the point, but I couldn't really find a good one. If I find one, I will post it on Blackboard. Okay? All right, so now we have to move to the next chapter, which is our DNA chapter. Ivan, you had a question?
Juan had a question before me.
Oh, I see that. How long is my expectation? You know, it's really hard to say. Like yesterday I felt, I'll tell you the two things I'm thinking. Yesterday I felt really hopeful because it felt like in New York the numbers were kind of going down. And so I was thinking, yay, but I don't know if you've looked at the numbers today, but they're way higher than they were. They're even higher than they have been. So I'm thinking probably June or July, that really till we can really go out and be able to mingle among each other and be close. If they can get this whole antibody testing ahead of time so that we can actually see whether or not we've been exposed, because some of us might have already been exposed and we just don't know it. I kind of have this feeling that we're all going to end up getting it one way or the other. At some point, you might have to get, we're going to be exposed to it. So I'm sorry about your 21st birthday. You'll just have to celebrate when you're 22. What are the numbers? It's something like 1,400 already today have died. So, and I'm looking at the death numbers. So let me tell you the other thing that I'm hopeful for. My husband keeps telling me I'm crazy, but I can't help it. I'm an optimist. I'm hopeful in our area that maybe we self-distanced early enough to reduce the spread. So if you look at North Texas, you'll find that we do have cases there, and they are going up. They're not going up very fast, But what I'm looking at is not number of cases, because I don't know whether you've looked, but in Fort Worth, in Tarrant County, they're not really testing very much. You pretty much have to go to the hospital to get testing. I mean, go to Dallas or something, but they're not testing very much. So the fact that the numbers aren't really high could be misleading. But what's hopeful to me is the number of deaths are really low. So for the number of cases that we have, we don't have that many deaths correlating with it. So I don't know what that really means, but I'm just hopeful that we've tampered it down enough that we're not spreading it right now. So the people that are infected are quarantined already, and they're not spreading it to new people. That's my hope. Okay, so I have some questions. Let's see. Worldometer is really great. That's what I've been using to keep track. You can go, if you click on like United States, it's a hyperlink. It'll take you to each state so you can see. And then the Tarrant County Public Health has like every city in Tarrant County broken out. So like I know for Keller, yesterday there were 13 cases. So are cytokine storms still a danger like the original Spanish flu? Yes. So absolutely. If it mutates, and remember how natural selection works. When the environment changes, we're going to select for those that are able to survive the changed environment. So once we introduce the vaccination, any virus that has mutated that can survive beyond that vaccination will continue to spread. So it's just natural selection at its best. And so once we really come out with the vaccination, we might really see that second resurgence of likely the mutated version. And if we don't have the vaccination in time, we might have that happen just because we all start to kind of go back out and interact. And if some of us still haven't been exposed, we can then spread it. All right, Ivan, you had a question.
On your YouTube, you didn't have a video on chapter 13. Is that going to go on later, or?
Oh, it's there. It's there. They just take forever. Like once I do them, they take forever to download on my computer, and then I have to upload them onto YouTube, and they take forever. So did you check today?
No, I didn't.
Okay, I think it's there now. I did it last night, but it was sitting there in like processing mode forever. So I think it's there. I'll check and make sure. If it's not there, I'll re-put it up.
Okay, I'll check right now.
Okay, so this next chapter is on DNA and DNA replication, really. I love the history of this. I'm going to do this very briefly. I won't test you too much on the history except for the part that involves the viruses, okay? But what's really interesting is if you think about the late 1800s, so that's when Darwin had gone on his trip, and that's when Mendel was figuring out the genetic laws of genetics. At that point, we really weren't sure what was the genetic material. It was pretty obvious that something was being transferred from one generation to the next to make them look similar, but it wasn't sure what it was. And so there were some pieces of evidence that suggested that it was either DNA or protein. So in 1819, a guy named Weissman, they had done some studies on some cells, and they thought that the nucleus seemed to be what controlled the traits. So if they somehow messed with the nucleus, the traits changed. So that suggested, since there were proteins and DNA and RNA in the nucleus, that something within that nucleus was involved. So that kind of led us to thinking DNA or protein. And then in 1900, Thomas Hunt Morgan, who's a huge fruit fly guy, he discovered a lot of cool things studying fruit flies. But one of the things that he did was he could study the chromosomes of fruit flies. So fruit flies are really cool. They have, in their salivary glands, they have chromosomes that instead of just, so when we learned about in S phase, how we make copies and then we have 2 of every chromosome that are in the X format, the duplicated chromosome format. Well, in salivary glands, they make thousands and thousands of copies of each chromosome. So they become these really thick chromosomes that you can see under a light microscope. Like you can just see them under the microscopes we have in lab. That's how big and thick they are. And so because he could visualize them easily under kind of early microscopes, what he would do is he would look for differences in these fruit flies. So normal fruit flies have red eyes, for example. He would find a fruit fly with white eyes and then study what the chromosome looked like in comparison between the red-eyed one and the white-eyed one. Normal fruit flies have straight wings. He would find ones with curly wings and then compare these. And so what he was able to do was chromosomes have that very unique banding pattern. I think we saw pictures of it at some point, but let me just show you a picture just for fun. Fruit fly salivary gland chromosome. Okay, so they have really unique banding patterns. Can you see this picture? See how there's like dark stripes? This is not something they wrote in. This is just what it looks like under the microscope. They have these very distinctive striped patterns, okay? And so he was able to look at differences in that striping pattern and determine that when he saw certain traits, so for example, if he saw long antenna versus short antenna, he could see a change in that stripe on one position of the chromosome. So that suggested that the chromosomes were influencing traits. Does that make sense? Okay, and so that they knew at that point that chromosomes were made up of DNA and proteins and sometimes RNA. So that really led it not just to the nucleus itself, but really more specifically to DNA or protein. All right, now here's a really cool experiment. This was started kind of in the early 1900s. People really started to want to know, what is it? What is this genetic material that's being passed down? And the thought process was that it was likely proteins, because proteins had seemed to have more versatility, right? They were enzymes and antibodies and membrane proteins and structural proteins, all those different kinds. So it seemed like that would make more sense. We know differently now, but that's what they thought at the time. So this was a really cool experiment, and I like to walk through these experiments because it's worth your seeing how experimental design is critical, okay? So they were studying two different strains of Streptococcus pneumonia. One of them did not make a capsule, and the other one did. So remember, we talked about that not all bacteria have capsules. Those that do make the sticky ooey gooey layer, and you can actually visualize it here, that makes it so that our immune system gets stuck and can't fight against it as easily. So the ones that have this capsule are far more likely to make us sick than the ones that don't. Okay, so this is a pathogenic bacteria, this is a non-pathogenic bacteria. All right, so they proved this. So they took the non-pathogenic one, which we call the R strain, put it in mouse, mouse was fine. Mouse immune system did its job. They took the pathogenic one, which we call the S strain, put it in the mouse, and the mouse died. So that just proved the difference between those two bacteria. So then they wanted to kind of discern what was making the mouse die, right? What part of it was actually doing this, okay? So so they were studying, they weren't really necessarily looking for the DNA protein thing, but this kind of came out as an aside. So what they did was they took the pathogenic bacteria and they heat killed it. So you know what happens. Heat something up, the proteins start to unfold, and if proteins aren't doing their job, then the cells can't do their work. And they took that heat-killed S strain and they put it in the mouse, and the mouse was fine, right? So if you're this kind of made it sound like, well, proteins are unfolding and they're not doing their job. Maybe it really is proteins, right? So this was the experiment that was pretty striking. So they took the heat-killed S strain, which remember the mouse lived, and they took the R strain, which remember the mouse lived, and they combined them together and injected them into the mouse. And the mouse died, suggesting that somehow something from the pathogenic bacteria was causing the R strain to become pathogenic. All right? So there was some sort of transforming factor. And so this is when they thought, here we finally have an opportunity to specifically hone in on whether it's DNA or protein. Okay, so this was the experiment that they did. They actually did way more than this. I'm just showing you kind of a little piece of it. So these first four things are the original experiment that they did. The R strain was fine, the S strain killed it. Heat killed S strain was fine, heat killed plus R killed it, okay? So then what they went in is they thought, we're just gonna we're gonna eliminate one thing at a time. So they eliminated lipids. They eliminated RNAs. So the question came down to, if we eliminate DNAs using an enzyme called DNase, which basically goes in there and chops up DNA into pieces, and we put that with the R strain, or we eliminate proteins we take an enzyme that chops up proteins in the heat-killed S strain and combine it with the R strain, what's the difference? So when they eliminated the proteins, it acted just like the original experiment. However, when they eliminated the DNA, then all of a sudden that heat-killed S strain could no longer transform the R strain, and the mouse lived. So that was really the first piece of huge evidence that suggested, oh my gosh, this DNA is important. And this wasn't really fully discerned until like 1934, okay? So all of a sudden in 1934, labs around the world were like, oh my gosh, we gotta figure this out. We gotta show this too. And we are now looking more specifically that it could possibly be DNA. So another lab that was doing work on the same thing, they were studying these bacteriophages, which we just talked about. And they knew that the phage was made up of just DNA encapsulated in proteins. So it seemed like the perfect opportunity to see what was really capable of infecting a cell, because obviously when the virus infects the cell, it transforms the cell and causes it to change. So this was done in 1952. I'm sure you've all heard of the Hershey-Chase experiments. This is probably the most famous one about determining DNA was a genetic material. So what they did was they used radioactive isotopes, so going the whole way back to chapter 2, right? Radioactive isotopes to distinguish the protein part of the virus from the DNA part of the virus. Seeing if they could separate those out in some way, could they determine what part was getting into the cell and actually infecting the cell? Now, luckily, they were using the phage, because we just learned that when the phage sits on top of the cell, all the protein particles stay on the outside and all the DNA gets squirted in. If they had chosen another virus, they wouldn't have been able to discern one from the other. So again, sometimes luck just plays into your success as a biologist as well. So they labeled the DNA with radioactive isotopes. They labeled the protein. And so what I mean when I say labeled them, they took virus, allowed it to infect a cell, and they only provided radioactive phosphorus, or they provided radioactive phosphorus. So when the new DNAs were being made from that viral infection, those DNAs had radioactive phosphorus incorporated into them. Then in a separate experiment, they took the virus, they let it infect the cells, and they added radioactive sulfur, because some amino acids have sulfur, so any amino acids that were put into these proteins that had sulfur would be radioactively labeled. And then they took those two pools of virus that they created that were each radioactively labeled and then compared them to each other. So they took the radioactively labeled protein and in a separate experiment, they took the radioactively labeled DNA, allowed each of these to infect the cell, right? And then at the end, they spun the cells in a centrifuge, just like we did in lab. You get a cell pellet at the bottom, and then you have the supernatant on top. All right, so Tuesday, Thursday, people, did you guys learn what supernatant is? Yes, no, maybe so?
No.
No? Okay. So when you spin cells, the bottom of the anything that's heavy, it's kind of like spinning with a bucket of water, right? The heavy stuff goes to the bottom of the bucket. So when you spin cells, the cells are all going to be heavy, and they're going to pellet at the bottom of the cell. And then anything that's not part of the cell will just be in the fluid above it. That's called the supernatant. So they did that. They spun it. So they had cells at the bottom and then liquid above it. And then they detected where they separated the cell pellet from the cell supernatant, and then they used means to figure out where the radioactivity was. So in this example, they found that all the radioactivity was in the supernatant, and in this activity, they found that all of the radioactivity was in the cell pellet. So that suggested that the DNA was actually going into the cell and causing the infection to happen, causing more virus to be made. So somehow the DNA was leading to new proteins, even though the proteins didn't get into the cell themselves. Pretty cool experiment, right? So this really led to the conclusion that DNA was the genetic material. And what year was this? 1952. All right, so like my mom was born in 1952. So this wasn't that long ago. My mom's still alive, right? This wasn't that long ago. So this really was the kind of the icing on the cake. There were labs around doing other things, but this was the icing on the cake of that DNA was the genetic material. Okay, some other things that were found along the way. Another group did some experimentation where they were looking at DNA across species. So they looked at, let's just say, horse and pig and yeast and E. coli. And they were looking at how many of the building blocks were present in these cells, right? So they would, count the number of A's, C's, G's, and T's. Now they could do this back then. They didn't know the structure yet, but based on chemistry, they could tell the difference in weight between these difference in mass. So what they found was that every organism had a different number of A's, C's, G's, and T's. But what Chargaff found was that in any species he looked at, the number of A's always seemed to equal the number of T's, and the number of G's always seemed to equal the number of C's, right? So we know now that's because of the complementary base pairing, because they pair up in that way. But this was kind of what led Watson and Crick to that idea of complementary base pairing. This is just such a cool picture. This is a real micrograph of a bacteriophage squirting its DNA into the cell. I just think that's the coolest picture ever. So I just had to show it to you. Okay, so you don't have to actually do this, but think about the general structure of a nucleotide. You know what it looks like. Could you tell the difference between an RNA nucleotide and a DNA nucleotide? And think about this three-dimensional structure of DNA. So the general structure of a nucleotide looks like this. Hopefully that's what you were picturing in your head. Tell me, if I were to, can you tell by looking at this whether this is DNA or RNA?
DNA.
Okay, how do you know it's DNA?
Deoxyribose. RNA has the 5-sugar.
Well, they're both 5-sugar, but deoxyribose doesn't have an oxygen right here, where ribose does. What's another way you can tell? Yes, good job, Rock. There's a thymine there, right? So DNA has thymine, RNA would have a U right here. Okay, so the next thing that needed to be determined, so now labs across the world, starting about 1950, labs across the world were like, oh my gosh, we got to study DNA, because nobody was studying DNA at this point. Nobody was. Nobody thought it was important yet. It's kind of not as easy to see and manipulate as proteins were, so really labs weren't really studying it. So across the world, labs are like, oh my gosh, we've got to study DNA. So people were doing all kinds of different things to try to figure out what the DNA structure was, how it looked, how it functioned, to put more meaning to this idea that it could be the genetic material. And so one group that had a technician named Rosalind Franklin on their team. She was a really skilled X-ray diffraction technologist. Her job was to do X-ray crystallography with stuff. Basically, when you do X-ray crystallography, you take a substance, you dry it down, you almost make a crystal of it, and then you beam electrons at it. And you beam X-rays at it, and then the X-rays are going to hit it and bounce off of it at various angles, and then you have a piece of film on the other side, and the X-rays make a pattern on the film based on the angle at which they're bouncing off. It's a very complicated, you have to be very skilled to be able to look at what you're seeing and make any meaning out of it, okay? So she was tasked with taking DNA that would have been excised from the cell and crystallizing it and doing X-ray diffraction, and this is the pattern that was on the film. All right, so it does look somewhat circular. It's kind of uniform in its diameter on either side. Kind of looks like some things are coming towards the middle, right? So there was a very distinctive pattern. So Watson and Crick were the scientists that were trying to deduce the structure of DNA, and they were using Rosalind Franklin's work and Chargaff's work and other things they knew about the chemistry to try to figure this out. And so they were trying to figure out, first they could see that it looked like it was double. It looked like there were two of them, not just one of them. So they were trying to figure out if there were two of these strands of DNA, because they didn't even know it was a double helix at this point. If there were two of them, how would they fit together? And so by looking at this pattern of X-ray, they thought, well, what if the purine matched up with the purine, or the pyrimidine matched up with the pyrimidine? That would give us the diameter of that helix wouldn't be right. So based on what Chargaff said and based on the X-ray data, they suggested maybe a purine was pairing with a pyrimidine, right? So an A with a T or a G with a C. So they looked a little bit more at the chemistry. Could that really work? Well, we know now there's possibility for hydrogen bonding between G and C easily. 3 of them between A and T easily. They form that way. They are very consistently complementary base pairing in this way, right? So what Watson and Crick did was they used balls and sticks to represent the different atoms, and they started just putting it together. They knew a little bit about the general structure of a nucleotide based on chemistry, and they were trying to figure out how they all connected together. And so you can see them working on it here, and they determined, you can see here the deoxyribose sugar. These guys see it there, right? So they were able to determine that it couldn't be just a straight ladder because things would run into each other, so it has to twist to give space, for example, for something sticking up right here not to bump into that. So it had to turn in this way so things had space. And so they ended up discerning that the backbones of our DNA molecule were a repeat of sugar, phosphate, sugar, phosphate, sugar, phosphate. The bases stuck in the middle. There were 2 of these sugar phosphate backbones, and the bases held those 2 strands together. They also were the ones that discerned that the strands didn't run in the same direction, that they actually ran in opposite directions, right? Which is called anti-parallel. I think we talked about that last time, that they run in an anti-parallel direction. We always label the ends based on the carbon numbers of the sugar, right? So when the O is pointing upward, the 5th carbon, which is like a chimney carbon, is pointing upward. And the 3rd carbon, which is this one right here, which is more like a basement carbon, is pointing downward. So in this strand here, the top is the 5', all the 5s are pointing up, and the 3 is the bottom, all the 3 are pointing down. And if you look at this other strand, it's the opposite. Here's my chimney, here's my basement. All the 3s are pointing, in this case, up, all the 5s are pointing down. And that does come into play and becomes important with DNA replication. All right, I have like 6 more minutes, so I'm going to go as long as I can. So those of you that have lab, make sure you pop off right at, you have lab at 2? Yes? If you do, make sure you pop off right at 2 and don't be late for lab. We'll stop right at 2 too, so you won't miss any material. All right, so how does DNA work? Well, we already talked about that it has to happen for the purpose of mitosis. So what is the purpose of mitosis?
To split membrane.
Well, not quite.
Cell reproduction.
Close, but what's the real purpose? I mean, can we just do it willy-nilly? What do we have to come up with at the end? So it's the division of the nucleus so that we end up with what? 2
Identical.
Daughter cells.
identical. That's the whole point. We need genetically identical nuclei and daughter cells, right? So during the S phase of the cell cycle, we're going to undergo that copying process of the DNA so that we have a copy, an exact copy in each cell. All right. So that copying process that happens in S phase is called DNA replication. It's very important that you don't mess this up with transcription. They're very similar in a sense, but they are very different, okay? DNA replication only happens during S phase, and that is only when the cell has committed to dividing. So in some cells, it happens very rarely. Other cells, it happens all the time. Transcription happens in all cells all the time, pretty much, right? Okay, and then Watson and Crick suggested there was a base pairing mechanism that was used for this copying purpose. What do we call that base pairing? Complementary base pairing, right? So we're complementary base where the A's always pairs with T's and the G's always pair with C's. All right, I'm gonna skip that question. So here's the basic gist behind DNA replication. First, the original DNA molecule in the cell, those are called the parental strands, okay? They have to separate from each other. So we're gonna have to break hydrogen bonds in here. In lab, we use temperature to do that. In the actual cell, we're going to use an enzyme, all right? And then each of these parental strands are going to serve as a template so that when the polymerase reads along, it knows exactly what nucleotide to put where based on the rules of complementary base pairing. So if you look here, the original parent was ATCG. This one is AT, CG, CG. Everybody see how they're identical to each other and identical to the parent? Yeah?
Yes.
All right. So this is called semi-conservative, which refers to the fact that the new DNA molecules have one old and one new, one old and one new. You can read in your book kind of the experiments that they did that suggested this was the way it was as opposed to two olds going together and two news going together. They also used radioactive isotopes to figure that out. All right, now how does this work? Well, there are places along the DNA where replication starts. So you might think, well, why doesn't replication just start at the end of a chromosome, of a linear chromosome, and just go the whole length? Well, you have to think about the origins of where replication started. So the idea of replication began in bacteria where there were no ends. There were no ends of chromosomes because the DNA is circular, right? So there had to be a region in the circular piece of DNA where it made sense for this to start. Typically, it's a region that's really high in A's and T's because there's less hydrogen bonds and it's very easy to separate the two strands at that location. All right, so this is called the origin of replication. And what happens is the two strands separate just locally in that region, and the whole idea of replication begins proceeding in both directions from that origin on both strands. So you can see here the old DNA, the parental DNA is dark blue and the daughter DNA is light blue. Okay, so it continues to happen until they basically meet at the other side, opposite side of the origin of replication, and you end up having two. Me still?
Okay, you're back.
Okay. Instead of just one, but you can see that they're identical to each other. Okay. On a linear chromosome, because of the way linear chromosomes are, oops, I think I have to start recording again. Well, I only have a minute left because linear chromosomes are the descendants of circular chromosomes, right? They don't necessarily have their origins at the end because wherever the chromosome, the circular chromosome broke to make the linear chromosome, that's how you're going to start. Can you guys hear me?
Yeah.
Okay. So the origins are not necessarily at the ends, they're just along the chromosome at various regions, but it happens in the same way. Okay? So we will start here with enzymes on Thursday. Okay? So I know you guys, oh no, can I go for 10 more minutes? Do you guys have to go to lab right now? Or does lab start at 2? What time does lab start? Somebody tell me? All right, I'm going to go for a couple more minutes then. All right, so the enzymes, obviously, that are involved in this, there's a few that I want you to know. The enzyme that separates the two strands from each other is called a helicase enzyme, okay? And the helicase enzyme basically goes in at the origin and separates the strands from each other. Now remember, they like to complementary base pair, so they want to come back together. So once we separate them, we have to make sure they maintain being separate so that we can get an enzyme in there to read them. So we rely on these single-stranded binding proteins to maintain the single strands of DNA. This one is harder to show you without being in class with you, but basically when we start to unwind the helix in the middle of it, the ends of it kind of get overwound, they get supercoiled. And so an enzyme called topoisomerase is going to go in and break the DNA, unwind it, and then put it back together to keep from overwinding. And then if you remember from lab, the DNA polymerase is pretty persnickety, and it can't start building a DNA strand from nothing. It has to have a starting point. It has to have a nucleotide already present. We used primers in lab that were complementary to the DNA. Well, in the cell, we have to build those primers every single time we need to do it. So the enzyme that builds those primers is called primase. And so what it does is it comes along and it builds a short, less than 10 nucleotide RNA strand along the DNA to give a starting position for the polymerase. All right, and then what the polymerase is going to do is it's going to read along the parent strands. It's going to start here. It's going to read along the parent strand and add in new nucleotides as it gets to each new nucleotide to build that new strand. All right, so real quick, I'm going to keep you for 2 more minutes. What kind of reaction does DNA polymerase do? It's putting nucleotides together. So what do we call those kinds of reactions?
Catabolic?
Not catabolic. The other one.
Anabolic.
Anabolic, right? It's going to be endergonic. It's going to be dehydration, synthesis, or condensation, right? And then the DNA polymerase is going to form a bond between the neighboring nucleotides. What kind of bond do we have between neighboring nucleotides? What connects the backbone? Oh, the backbone. What connects the backbone? The sugar phosphates. What kind of bond? I'm still waiting. I haven't seen the right answer yet. Not hydrogen. Hydrogen is what connects the two in the middle. But when we're actually building a new strand, it's not an ester bond. It's a covalent bond between the phosphate, I'm trying to give you a hint, and the sugar. So it's a phosphodiester bond, right? It's a phosphodiester bond. Okay, so those bonds are going to connect the sugar and the phosphate. All right. So if we think about the fact that structure reflects function, the polymerase has to have the right structure to do what its job is. So it basically is like a donut or a clamp that clamps onto the DNA to read along it. And here's where I'm going to stop to let you think about this. Now, the DNA polymerase in our cells, in all cells, is very persnickety. It has a couple of situations that have to be just so for it to work. One is it has to have a starting nucleotide, which is why you must have the primer first, because it can't initiate synthesis from nothing. So it has to have a nucleotide to add it to. The other thing is that it only works in one direction. All right. So when it's reading the parent strand, it can only read the parent strand from 3' to 5'. All right, it can only read in that direction. It can't go down here and read up this way. It can only read 3' to 5', which means, if we go back, that the new strand is made in a 5' to 3' direction. So I like to think of it just how do we read it? It's always read 3' to 5'. That way we can infer how the new strand is being built. All right, so it always reads 3' to 5'. That means the complementary strand is going to be opposite that. Okay, so now I think I'll stop and let you guys have at least time to go to the bathroom before you have to go to lab. So we will start here next time. And probably what I'll do is, if you guys could read this before you go, I'll probably do this part with a video. I'll talk a couple slides through and then I'll show you a video, which really makes it far more easy to understand. Ivan, did you have a question? Ivan? I can't hear you, Ivan.
Sorry, I disconnected.
What's your question?
Hello?
Yeah, are you there?
I couldn't find the chapter 13 on YouTube.
You just looked right now?
Yeah.
Okay, I will, maybe it pooped out on me mid-processing, so I'll reprocess it today. I'll put it on there.
Okay, thank you.
Okay, so I'll see you guys on Thursday at 12:30, and then I'll be on here for the next hour. I have to log off for a minute, switch rooms, because my husband likes to be in this room for his class. So give me a couple minutes if you come in. I'm not there, just wait. And I'll be in office hours from 2 to 3. Okay, stay safe. Don't spread it. Bye, y'all.